Fran, thank you so much. It was great to actually meet you and match a face with all the emails that went back and forth. And thank you to Sandy for the cookies. Important to have cookies. But it's really great to be here. I have tried to make it a point to get out and meet as many folks as I can, because I think it's always important to let people know about all the exciting things that we're doing, and also hear some of your concerns. So I like to do things kind of informally, and if anybody has a question, I always find that everyone gets more out of it when it's a back and forth informal thing. All right, so I'll get started, and I'm just going to hit a whole spectrum of things that I'm almost certain that you're all interested in. How do we get this? How do we get things like Alzheimer's? What do we know about it? What can we do about it? I may not hit everything, but if I don't hit something that you really want to know, raise your hand or send me an email if you remember it later, okay? All right, so we'll get started here. So this is my group. I have a pointer. So this is good, so I can point here. This is my group. We're at UNC, at UNC's campus in Chapel Hill, and we have quite a vibrant bunch, as you can see here. And I'm in the Department of Neurology, and if anyone's interested, oh, thank you. If anyone's interested, there's more information up on this table for things we work on, my card, and other things like that, okay? And by the way, this background here is an Alzheimer's brain. And so we'll get into this in a few minutes. But every little blob that you're seeing here is a tangle that might accumulate in the brains of Alzheimer's patients. And so this is what we want to get rid of. And so using this as my background slide always helps remind me about the thing that we want to get rid of, which is these tangles that accumulate in our brains. Okay? All right, so just a little bit about me. As Fran mentioned, I grew up in the best city in the world, Philadelphia, Eagles fan. And I grew up near I-95, and I only mention that just to point out something that I'll tell you about later, that there is an interesting risk for growing up near pollution and near highways. So there is this a new negative risk factor, positive risk factor, where you're increase your chances of dementia if you live near highways, which we'll get to in a little bit. I went to Penn State as an undergraduate. I worked at the Penn Staters are out there, very nice. I worked at the Fox Chase Cancer Center, also in Philadelphia. And then I discovered North Carolina and what a beautiful place this is to live. And so I went to Duke for my graduate training, and then I went back to Philadelphia and was at UPenn. And then I decided that the Blue Devils weren't my thing, and so I joined UNC and became a Tar Heel in 2014. So that was my trajectory. And initially I was interested in cancer, but I couldn't quite figure out cancer because it was too complicated. And then I realized that if we could understand what these plaques and tangles do in the brain, that I could actually contribute, and it was a tractable problem. So we got very interested in Alzheimer's disease, and so that was how my life changed. I also have a grandmother who passed away from Alzheimer's disease, so these things led me down the path where I am today, okay? So this is my grandmother. She passed away about 10 years ago, and her name was Jewel Brody. I watched her progressively over the course of about 10 years start to show things like gait disturbances, physical deterioration. She lost weight, her posture became quite deteriorated, mood disturbances, irritability. This was a person who never said a swear word in her life and all of a sudden became the most, inappropriate person that you could ever be around. And so these were all indicators that something was not quite right. Okay, and so when you couple this, an unexplained disease, with the fact that nearly 100% of all drugs failed, including the drugs that she was on at the time. I'm sure many of you have heard of Aricept and another drug called Namenda. These drugs are commonly given to dementia patients, and although they can improve quality of life a bit, they don't do much to the course of disease. And so I was confronted with this Interesting problem, and how could I start to, make progress? And so that was where my calling came in. So here are the things that keep me up at night, quite literally. Why do some of us get Alzheimer's disease and others don't? Okay. How do our genes determine whether we get Alzheimer's disease? What is it about our genetics that might predispose you to get this disease? What is it about aging that leads to Alzheimer's disease? No children get Alzheimer's disease. This is a disease of aging. So what happens to your brain as you age that makes you at increased risk? Age is the biggest risk factor for Alzheimer's disease. Without this time component, we wouldn't get it. So people don't talk about that too often, but what does the time part do to your brain? It's something we've gotten very interested in. And then lastly, and I just realized these are all apparently number one. That means they're all important. And then the last and most important is, can we develop new approaches to beat this disease? And I'll show you a couple that I think might look very promising, okay? All right, so this, yes, question?
Could you add to that question, why are 2/3 of Alzheimer's patients women? Why are women more predisposed to this disease?
Yeah, it's a great question. It's something we still don't quite understand. There's a lot of hormonal influences on the brain that people are now trying to sort out. And so there could be sort of male-female hormone differences that might regulate the brain. But the short answer is people still don't know. And women are certainly more at risk. And it might have to do with the risk genes also influencing hormonal status. So there's a combination of your risk factors, male versus female, and then the onset of disease, which is still not clear, but it's a very good question. Still don't understand. Any other questions? Okay. So I show Uncle Joe. So this is my wife's distant relative, who we call Uncle Joe. Joe Newman lives in Sarasota, Florida, and Joe he's about to turn 107, okay? Joe's wife, Anita, is about to turn 100. So between the two of them, they are 207 cumulative years. And I use Joe as an example of healthy aging. He's someone we want to strive to be like, right? And so instead of studying Alzheimer's disease by studying the ones who get it, you could flip it around and say, why don't we study the people who don't get it? And so Joe still drives. He just bought a Lexus and a Mercedes. He still reads. He's an avid reader. I even think he still dances with his wife. And so this is really an amazing story. And if anybody looks him up, he ran for Congress not too long ago. This was about 5 years ago, and did quite well, but wasn't elected. And so Joe, I've interviewed Joe, and so I want to understand what it is about his brain that creates this resiliency. Okay, but I'm happy to talk about it in the question session. But I just want to bring this up because this is a great example of what we should strive to be like. How has his brain avoided all these hallmarks of Alzheimer's disease? No evidence of dementia whatsoever. And so this is something that we all need to start to think about. How do we achieve this? Sorry?
I want to know about his wife's brain.
About his wife's brain. So this is what's going to happen if we don't act sort of immediately. So here we are right here. There's about 5 to 8 million people in the country have Alzheimer's disease. There's about 350 million people in the country. So of the 350 million, if 8 million have Alzheimer's disease, that's a pretty sobering reality, right? And so this is the trajectory. By 2050, you're looking at about upwards of almost 14 million people. And so this is broken down by age group. In other words, the older people are going to tend to get, the more elderly are going to get Alzheimer's disease at faster rates. And so what's going to happen is the elderly population is going to be more at risk for Alzheimer's disease unless we find drugs that can actually halt it. Okay?
Question.
Yes, question.
Do we find more of Alzheimer's in the U.S. than other parts of the country, or is it about the same statistically everywhere?
You set me up perfectly, because I'm going to show a map of the U.S. in a minute, and you're going to see a very surprising result of whether we get it equally in all states in the country. Okay? So hold that thought, okay? So this is Dr. Alzheimer himself. Sometimes I wonder what this man would have thought 115 years ago when he discovered the first patient ever that had Alzheimer's disease, who he coined. He called her, he didn't call her an Alzheimer's patient, he called her a dementia patient. Her name was August Dieter, and she was frantically running down the halls of her apartment complex, and she was starting to develop pretty severe Alzheimer's disease. She was cursing, she was screaming, she kept thinking people were after her, and he took out, when he had access to her brain tissue, he characterized it, and he was the first man, and she was the first brain sample to ever be shown to have what became known as Alzheimer's disease. And this is what he found in her brain. This is a tangle. This is an accumulation of junk in the brain that is unable to be cleared. And it's everywhere. It's everywhere. And so this hallmark is thought to be the thing that contributes to the neuronal cells dying in the brains of Alzheimer's patients. So a lot of people agree if you didn't have these, you wouldn't get the disease, which means we need to figure out how to get rid of these, right?
What is it?
So this structure is an accumulation of a protein that normally is benefiting the brain, but somehow starts to clump up and form we call it an aggregate, which is just a clump of garbage that needs to be gotten rid of, but your brain has lost its capacity to get rid of it. And so it just starts accumulating. And it accumulates in all kinds of forms and sizes. And if I had a slide with me right now, you could look at a brain of a patient that had this disease, and you would see that the brain is littered with these things. And one other thing I should tell you is that everyone in this room has some of these. The older you are, the more of them you have. Now, some people, like probably Uncle Joe, have a resiliency that probably keeps these things at bay. But other people are unable to do that, and why that is not clear. And it's going to vary from person to person to person. Okay? And so we need to figure out why they form and how to get rid of them in people that have a lot of them.
It only shows up in an autopsy, like an MRI or PET scan.
That's exactly right. But now, more recently, we've got PET imaging to detect these in live people. And this is all still early days, but you can get an injection, an IV injection, and these things can light up in the brain of live people. It's still crude, but it's going that direction. Now, the question is, if you could be told you had these things in a live person, would you want to know without anything? I heard some groans and mostly no's. And I probably would agree. I don't know that it's all that helpful to know that information. Some people might disagree and want to be forewarned when something like this was sort of creeping through the brain. But we don't have anything we can do at the moment, and Aricept certainly isn't going to get rid of those structures. Okay? So, yeah, question?
What is the young age of the youngest person you were ever found in?
That is also a very good question. These can be found in very young children. So these can be present in children. And there are some people that have early onset Alzheimer's disease. And so you might have heard stories of people in their 20s or 30s. And in those instances, these start very early. And so you could imagine a 10-year-old with these structures. Now, that doesn't mean they have Alzheimer's disease. That just means they're starting to accumulate. And in fact, in very young children, these have been detected. Mostly restricted to certain regions of the brain, but these are present in all individuals, and the older you get, the more they increase. Yeah?
Were those children tracked to see if many years later anything was developing?
Well, remember, you couldn't do live, there were no PET tracers, so you could only see these things with post-mortem end stage, autopsy. Now we can do PET imaging, and so maybe children being imaged today, we could follow them over the next few decades and see if they develop symptoms. But remember, children don't usually, even the early onset cases don't start until their 30s, maybe? Question?
Do people with progeria get Alzheimer's?
I don't know the answer to that, but there are other diseases that are at, the question was, do people with progeria? I think in progeria, everything about you just ages at an elevated pace, but I think you can age successfully and you can age unsuccessfully. Alzheimer's is considered unsuccessful aging. You have aged pathologically. And so I would separate, in my mind, and other people might view this differently, I would separate progeria and Alzheimer's disease. Now, other diseases are at risk for Alzheimer's disease, things like Down syndrome and things like diabetes and obesity. Those are two instances where your risk of Alzheimer's is higher. But progeria, I would probably say, falls into a different category.
What about amyloidosis?
Say one more.
Similar.
Say one more time.
The plaque.
Oh,
Amyloidosis.
that's right. That's right. So I'm actually going to show a picture of that in a second. Let me get to that picture, and that might clarify here. So this is an actual brain of a normal person, and this is the brain of an Alzheimer's patient. So this patient actually lost about a third of the mass of the brain. You don't have to be a scientist to make any, educated guesses about which one was the normal. This is a substantial loss in neurons. So if a brain weighs 1,500 grams, the Alzheimer's patient weighs about 1,000 grams. That's a loss of brain. So actually, when you think of it that way, it's not too surprising, right? You're losing function, brain function, quite literally, okay? And then to get to your question, this is what an amyloid plaque is. And I just want to clarify these terms because they're important. When a person has Alzheimer's disease, they have two major hallmarks. One is inside the neurons. That's called the tangles. That's what these are, what I've just been talking about. And then the other one is outside the neurons, kind of floating around in the space outside your actual neurons. Those are called amyloid plaques. And when you have Alzheimer's disease, you always have both of these. And what they think is that these came first. These led to these. But it's not these that end up killing the brain cells. It's actually the tangles that end up killing the neurons. So people have referred to this as the trigger and the bullet. Okay, does that make sense for a morbid analogy here? Not to get too morbid, but I think that it's an appropriate analogy. If you grow these in the brain, outside the brain, then what ends up happening is inside, these things have a higher tendency to form. These end up killing the cells. Yes?
Does MS lean towards Alzheimer's?
I'm not aware of any risk between MS and Alzheimer's. No, I'm not aware of it. Question, yep. You're next.
Are these living slides or are they post-mortem?
Oh,
Somebody-
sorry. One in the back and then right here.
My stepmother had alcohol-induced Alzheimer's.
Uh-huh.
Is that different?
This is a very hot topic right now, whether alcohol, so the question is alcohol connected to Alzheimer's? And I think the answer is probably yes. I don't know how, and I have to tell you, this is the newest thing right now. There's an actual request from the government to study this connection between alcoholism and dementia. And I didn't know until recently, but the data is pretty strong that they're connected. Question?
Are these living slides or are they post-mortem?
These are post-mortem.
Do you have living slides?
I don't, but
Are they available?
They are. You could search for them and get them. The problem is they're very low resolution and they're not clear because they're imaged through a brain on a scanner. So it's hard to get the clarity that you're seeing here. Do you see this? This clarity is quite high, and it's so precise. When you start scanning through a brain, it just loses all of the accuracy. And so that's why PET imaging has really not gotten to where we want it just quite yet. But it will be there eventually. Yeah.
So you mentioned PET scanning imagery. What about MRIs? Do they show any of these things when a person has an MRI?
Yeah, an MRI is complementary to this. But an MRI would just show you the volume of your brain. And so that could show you where structures have shrunk. But it wouldn't show you these things themselves. It would show you if part of your brain actually shrunk. It wouldn't
show the previous slide.
That's right. That's right. That's right. It wouldn't highlight these structures, which are the things that are doing the killing. Does that make sense? Mm-hmm. Okay. All the way in the back.
Is Alzheimer's hereditary?
Yes, Alzheimer's is hereditary, but it's a small fraction, a very small fraction, that have a familial component to it. Most people get what we call sporadic Alzheimer's disease, which means you couldn't trace it back to an obvious family history. But there is a small component where if you have the gene that causes it, you will certainly get Alzheimer's disease. It's just a matter of time. Those are the cases that are the most difficult to watch. Those are the ones that happen in their 30s and 40s, and then they pass it down to their kids, and then you see this string of siblings and kids that all have it. Yeah?
So is it a dominant gene?
It is a dominant gene. If you have the gene, now remember, it's only 5% of cases of people. The vast majority, 95% of people, get this sporadic Alzheimer's disease. And so we're only talking about a small percentage, and you can be tested. Anybody could have a test done on that particular gene. Okay? Question?
Can you see these tangles in the plaques with the naked eye?
That's a good question. You can. These are microscopic. You can't, but you can see the damage they do. They leave holes. They leave gaping areas where they've damaged. So you cannot. But if you dipped the brain in a dye that could light them up, then you would. You'd still have to look at it under a microscope to see these structures, though. Okay? Yeah?
And you may be going to cover this later, but what is the difference between Alzheimer's and dementia?
Yeah, and that's a great question because a lot of people think that they're the same. The reality is there are many types of dementia. There is frontotemporal dementia, which a lot of you may have heard. This is a different clinical disease than Alzheimer's disease. There's also Parkinson's disease-related dementia. There's vascular dementia. And so there's different types of clinical dementia that all fall under the umbrella dementia. Alzheimer's is one of them, okay? So there's like about 5 or 6 arrows coming out of an umbrella called dementia, but that's a very good question. All right. Keep them coming. I love it. Good questions. So this is the most sciencey slide you're going to see all night. Whenever you see a graph with X and Y axis, you know this is serious science, okay? All this slide is saying is that you have more dementia when you have more tangles. Did everybody get that? That's a pretty sciencey slide. That's the best way to say this. If you have lots of tangles in your brain, you are at higher risk of dementia. And they've done this by counting and then by taking the clinical tests that were done on those patients and seeing how they scored on memory tests. And so, of course, you have to count the tangles after they die, and then you section their brain. So that's how this was done. Painstakingly done, but very accurate. So we have gotten interested in how to stop this process. And at UNC, we published a study, and of course the popular press got a hold of it, and they give it names that are not always the most accurate. But you know how, an end to Alzheimer's disease. Scientists show how we could block the disease in its earliest stages. As much as we'd like to say we ended Alzheimer's disease, we didn't quite get there. But I'm just going to show you what we did find. We're really excited about a couple drugs that we found that we think might be effective. Okay? So here's an example. and I don't know if you can see this so well, but these are neurons that we grow in a dish. And what we do to these neurons that we can grow in a dish is give them Alzheimer's disease. We give them Alzheimer's disease by giving them inflammation. We all know inflammation is bad, right? We should limit our inflammation. Stay stress-free, do yoga, meditate, eat well, because if you don't, inflammation goes up. And when inflammation goes up, these neurons start to pull apart, and all these little blebs here are the remnants of these neurons dying. When we give these neurons a drug called TBST, all of a sudden these neurons are fully functional and nice and long, and they start to assume their little network of processes that go out and touch each other. These are healthy neurons. So this drug is good. This drug is going to help these neurons survive when you have Alzheimer's disease. This drug, called AP5, was amazing. This drug essentially kept these neurons fully functional, even when you were trying to kill them the best that you could with these inflammatory factors that are trying to damage these neurons. So it made them resilient. So this is just two examples, two drugs, TBST and AP5, that we hope will be in people someday, or drugs like these drugs. We hope they'll be in clinical trials in people. And so what we do is we start using experiments like this, where we grow these cells in dishes, and then we put these drugs into animal models like mice or rats. And so you're looking at an experiment done in cells, in culture. We grow them in a dish. We're now putting these drugs into mice. Yeah.
What time frame do you foresee?
To get these drugs into people? So, gosh, that is a good question, because the US seems to take a long time to do those things. And so from the point you discover a drug, it could take 10 to 15 years to get it into people. And that's one of the reasons why people tend to do some of these studies in other countries, where the administrative hoops that one must jump through are not as difficult. And so the rough time frame in the US is probably 10 to 15 years. But if you can show quickly that this works in animals and potentially monkeys, then all of a sudden you've got a line of 20 to 50 patients that might want to take this drug. And I had someone call my office from South Africa, Cape Town, South Africa, who wanted this drug. They wanted to be put on this drug. And I said, well, let's see if there's a clinical trial. And so we found a clinical trial in Germany where they were doing a clinical trial with a drug just like this one. And so they called and they got put on that clinical trial. And so they're currently taking a very similar drug to this one in Germany, in a clinical trial in Germany. Yeah.
What do those initials stand for, please?
Well, okay.
What type of a drug is
Yeah, and so this drug is a histone deacetylase inhibitor, an HDAC inhibitor, and this drug is an NMDA receptor inhibitor. Did that all make sense?
No, but
And so there was some reasoning behind using these particular drugs, because they were shown in previous studies to possibly do something good to neurons. And so I'm happy to send you information on these drugs.
Are they related to any drugs that we might know? Or anything
well,
that's their way out?
Yeah, and that's a good point. So this drug here is similar to a drug that is given to cancer patients. No one has looked at it in the context of neurodegenerative diseases. And this drug has not been given, to the best that I know, to people, although there is a version that is in clinical trials. Well, there is a drug called memantine that is currently given to Alzheimer's patients. Memantine does something similar to what this drug does, if that makes sense. Yes?
So these drugs are slowing the progression, they're not curing the disease.
They're not
getting rid of the tangles.
They're not getting rid of it, they're just preventing the damage that would occur from the tangles. So they're creating neurons that are super healthy, even in the face of the stress. Did that make sense? They're creating resiliency
In the remaining.
in the neurons these neurons have not died, so they're still healthful, they're still healthy.
Boosting.
It's
the ones that are
boosting
still
their resiliency. That's exactly,
fight off
that's right.
The deterioration from the tank.
And we think the tangles are creating an environment that's inflammatory.
Yeah.
And so we think this inflammation is actually possibly creating the bulk of the death that is coming from where the tangles are. And we think if you can make your neurons resilient, you'd be able to ward this off. Okay? Yeah. Yes?
So is there any testing of drugs for heart disease having some effect on these tangles? Are there any testing done
For drugs that are important in heart disease.
for heart disease and having a side effect
As a benefit to Alzheimer's disease.
as a negative?
Oh, as a negative. I don't know the answer to that, but what I can tell you is that when your cardiovascular improves, in other words, if you're taking a heart medication and you are benefiting, then typically your brain benefits. The heart-brain connection is very strong. So I thought you were going to ask whether that benefit would improve, but I don't know about drugs for the heart that actually worsen your condition.
How about cancer? Drugs for cancer?
Cancer is an interesting one because people repurpose those drugs all the time, and in a lot of instances, drugs that have already been FDA approved can then be tested pretty quickly in an Alzheimer's patient. And so there's a big push to take cancer drugs and test them in Alzheimer's disease because it's so quick and it can be fast-tracked. So some drugs, like this TBST, this is a drug that is given in a different form to people with T-cell lymphomas. And so you take this drug that's been effective in cancer patients and say, is it working for different reasons in an Alzheimer's patient? And it may. It may. Fran.
How about the new thing with Biogen, the drug that they're trying to get into people?
Yeah, Biogen has had ups and downs. You might see from the newspaper headlines, it's like every day a failure, and phase 2 trial failed, and then that trial that failed, they went back, they reanalyzed it, and it turned out there was some improvement with more numbers and more data. And so I think we just have to kind of hold out until, now, unfortunately, I know everybody wants to know, but the problem is there's so many people and so many data points, it just takes a long time to analyze. You hate to actually get it out there and be wrong. So they got it out there, they said it didn't look like there was any benefit, and then they did the analysis again, and lo and behold, it might actually have been effective. But I should say their drug targets the plaques, not the tangles. So there's a big push to understand how targeting the tangles is going to be more effective than targeting those amyloid plaques. Okay, it's a really important point because we haven't been able to target the tangles yet. We're now going to start to do that. Yeah?
Does somebody who has an inflammatory disease of another kind be more susceptible to tangles?
Oh, that is a good question, and it's a direct prediction of this, right? It's a direct prediction. And so I think it depends. What's the inflammatory disease that you're thinking of? Are you just saying in general?
Just in general, like rheumatoid or lupus or one of those.
Right. I'm not aware of one, of there being a direct connection. But there is a connection between diabetes and Alzheimer's disease. And diabetics have circulating inflammatory responses that are higher. And so there's all this direct or indirect. It's like what's direct and what's indirectly causing the Alzheimer's. So the short answer is I don't know, but I would say that chronic inflammation throughout decades is probably, this is just my personal opinion, is probably going to put you at risk for brain inflammation, which could tip you over the edge. I almost view it as you have a bucket, And when that bucket gets full, it can tip you over the edge and put you at risk. And so my personal opinion is that it would chronically over many, many, many years. But I don't know that to be the case. In the back.
What substance do you measure for inflammation?
Well, you can measure circulating factors that are in the blood or in the spinal fluid or in the plasma. Cytokines, exactly. Cytokines, interleukins, TNF, alpha, these are things that are marks of inflammation.
So
They,
when you do that and you add TDST or AP5, it reduces...
they go down. They go down, exactly. That's right. Yes?
Have you tested metformin?
You know, there was a time where we were going to, but we haven't done it. We have not done it. And metformin, yeah, this question was about metformin.
That's for anti-aging.
That's right, that's right. And there are a lot of people that take metformin in an aging capacity, totally independent of diabetes. Metformin could actually be a healthful drug. And I'm not promoting it, but there are a lot of people that want to live healthier, longer lives that are on a daily dose of metformin. Yeah.
Well, I know there's a thesis that Alzheimer is really a symptom of the greater disease, which is aging.
Yeah.
Treating aging as a disease.
Yeah, that's...
Is there a correlation or research on aging and Alzheimer and the emerging?
Yeah, I think that the aging brain tends to want to become an Alzheimer brain. And a lot of people think if you live long enough, you will eventually get Alzheimer's disease no matter who you are. In other words, Uncle Joe, who's 107, would eventually get Alzheimer's disease if he lived to 150. Unless you treat aging. And so I think you've got a controversial theory, which is how do you treat aging independent? A lot of people like to single out the tissue, the organ of interest. But if you view it as a whole and you just prevent aging, then actually all your tissues should improve. Every organ should get better. But targeting aging in Alzheimer's disease has not been something that's taken off yet. All right. So I think this is a good visual for what happens to your neurons when you age. Here's a healthy neuron. This is a neuron that sends an axon all the way down to another neuron. And all these little pieces of this neuron reach out and touch other neurons. This is a healthy neuron. This is an early onset Alzheimer's neuron. You can see it starts to shrivel up and lose some of these dendritic processes. And then in a later stage Alzheimer's disease, you essentially see what amounts to a dying neuron. And so this is what happens to the neurons. They pull their arms back, they start to shrivel up, and they die. Okay? And what's doing this is the tangles, the excessive inflammation, and the presence of the plaques, which happens earlier. So you have a triad of things: plaques, inflammation, and tangles, which are the three most obvious hallmarks of Alzheimer's disease. Okay? And so our approach has been to give mice Alzheimer's disease. And this is a bit problematic, especially when you have a mom who is in PETA. My mother is a big advocate for animals' rights, and she is in PETA, and so you can imagine how those discussions go. But giving a mouse Alzheimer's disease is now a reality. So we can take a normal mouse, we can put it in a pool, we can ask this mouse to find a platform, and we can train it to find the platform. And that mouse uses all the parts of its brain that it needs to find that platform, okay? And it does it in a way that mimics what happens in an Alzheimer's patient. Because if you give this mouse Alzheimer's disease, all of a sudden this little poor mouse can't find the target quadrant, and it starts to roam around the pool, and it never finds the quadrant. This is a submerged platform. This is a platform, it's like swimming in a pool to a little seat, and it cannot find that seat because it has tangles in its brain, okay? And so what we do in the lab is we give these mice little memory tests, and we ask, can any of these drugs make the mouse's memory better? Okay, and so what we're doing now is we're taking those drugs that I just showed you that worked in those neurons grown in a dish. We're giving these Alzheimer's mice those drugs, and preliminarily, they're getting better. And so this takes a long time. But it doesn't take as long as a human takes to get Alzheimer's disease. This mouse gets Alzheimer's disease in 1 year. In 1 year. And so in a year, that mouse will have impaired memory and it will slowly succumb. And so what we're doing is injecting those drugs into this mouse and asking if its memory gets better. And we think that with all the drugs we're using, there are going to be a handful that make that mouse better. And those are the ones we really want to study and get into people. So here's an example. So here's a mouse that had Alzheimer's disease. This mouse, when you test it over a period of days, it couldn't learn. It couldn't find this platform. As many times as it tried, it took the same amount of time to find this hidden platform, even though it should have been learning. If it learned, it would have found the platform faster, and this would have been going down. But it didn't learn, okay? It continued to have symptoms. This mouse, on the other hand, this mouse got an injection of a drug, and all of a sudden, it started to learn. And so by the end of this course of testing, this is 9 days of testing, this little mouse all of a sudden could find the platform in about 10 seconds. So we classify this as spatial memory and learning, okay? And so what we're doing is we're dosing mice with these drugs and asking them if they can learn better. And in this case, it looks very promising. So that's one example of a drug. And so what we hope is that potentially we can take that drug, we can package it into a formula, and we have ongoing efforts in our lab with various companies where we could potentially, at early signs of Alzheimer's disease, potentially dose people with drugs that look like they're either removing these tangles or increasing memory, or potentially both. And so first we have to start preclinically, and eventually we get into phase 1 clinical trials. Fortunately, UNC has quite a patient population that has early onset diagnosis of Alzheimer's disease. So the idea is that we could begin begin phase 1 trials with the patient population that's seen at UNC. And so all of that is happening within the next few months, and we hope to start enrolling people on drugs that become FDA approved. So I just want to highlight a technology for anyone that came here to hear all about the newest things in science. I just want to highlight a technology called Clarity. Has anybody heard of Clarity? Clarity? Okay, Clarity is the process by which you can take a brain and make it translucent. The same way that a jellyfish would be translucent. So this is a jellyfish. This man pulled this jellyfish, I don't know why, but this man pulled this jellyfish out and he's holding it. This jellyfish is translucent. So someone invented a technique that we call Clarity that can take a brain which is sitting right here with this yellow tinge, and you can see it's translucent because we can see the newspaper letters behind it. By making a brain translucent, I don't think people appreciated it at the time, we can now hold this piece of brain tissue up to a microscope and peer even deeper and with higher resolution than we've ever seen before. Now we can see things that traditionally would have been nearly impossible to see. So we call this technique Clarity. And I just want to bring it up. First of all, you'll remember it because clarity is a cool word. And secondly, you're going to see how this technique revolutionizes the whole field, because your brain is so dense with lipids and with things that make it impossible to see through. Now we can see through it all the way to the other side and see everything in it, plaques, tangles, and everything. And I think I have a video of this. I'm not sure if this is going to play, but let's give it a shot. Oh, it's working. Okay. So this is an Alzheimer's brain, a post-mortem brain from an Alzheimer's patient. Everything you're seeing in green is a tangle, and everything you're seeing in blue is a plaque. So you're taking a little bit of a ride through this piece of brain tissue. You can see how many plaques and tangles are littering this brain. It's just covered. So I'll play it again because it's... Let's see if I can play this again. And everything you're seeing here is a neuron. These are all neurons, tangles in green, and then you're going to see much less plaques in blue. Here's the plaques in blue. You can see them. They're bigger, but there's much less of them. Okay? So that's an example of what clarity can do. We can't do that. We couldn't do that until just 2 years ago. And so that's the advantage of having really smart people invent new techniques. Okay? And here is an example of a very bad inflammatory microglia, which is the immune cell in the brain, that is trying its best to reach out with its arms and grab all the little tau tangles in red. These little balls are tau tangles. If you look real close, you can see these microglial arms holding on to these red balls here and actually trying to pick them up, like this one right here, and eventually we think they'll engulf it and try to get rid of it. But what happens is, in the brains of people that have inflammation, these cells get irritated. These cells get irritated and then they start spitting out inflammatory factors that end up doing more damage than good. And so we think that's why inflammation exerts such a bad effect on the brain. Because you have an immune cell that goes in with good intentions, but eventually becomes irritated and starts to create havoc in the brain. And so this is a cell type of interest in my lab right now, to try to understand how these cells become dysfunctional. Call it dysfunction. Okay. How do we identify people that are going to get Alzheimer's disease? Well, fortunately at UNC, we have really good clinicians that do spinal taps, and they take spinal fluid. Remember, your brain bathes in spinal fluid. So we can take this spinal fluid out of the patient. We can take this, we can store it in the freezer, and we can use big fancy machines. This machine's called a mass spectrometer. This mass spectrometer can tell you every protein that's in your blood, your spinal fluid, your brain, with precision and accuracy that is unprecedented. So we're using this type of a machine in my lab now to try to identify biomarkers. What can we pull out of the spinal fluid or the blood that could tell me if you're going to get Alzheimer's disease? Because remember, we just talked about knowing whether you're going to get it could be an amazing thing. Because if we knew and then we had a drug, we could treat you before you even got the disease. That's what this machine and this approach is helping us do. And so here's an example of the data, and I'm sure all of you have seen this before. I'm just kidding. This is just a string of proteins here. This is amino acids that make up a protein, and every little tab here is a different modification on that protein. And so all I show this for is to tell you that we're able to do mass spec and identify all the proteins that you have present in your spinal fluid or your blood. And this is our validation of it. And where this becomes powerful is if I start taking a patient that progresses with Alzheimer's disease and I start to monitor this profile and say, well, it looks like you're progressing, if I had a marker that I could follow. And then if I could treat that patient and I could say, well, it looks like you're getting better because I have a marker that I can follow. Okay? So the power of a biomarker is not to be underestimated. I know we want cures, but we also want biomarkers. And so we need that biomarker so I can follow disease progression or disease responsiveness to a potential drug. Okay? And so you can imagine, if you had a particular mark in your blood or spinal fluid, say you had 3 different marks. we are imagining a day in the not too distant future where we could distinguish between whether you had Alzheimer's dementia, frontotemporal dementia, or progressive supranuclear palsy, which is another devastating dementia. It's a movement disorder. And so we think that our biomarker approach could allow us to distinguish between dementias as well as distinguish your stage of dementia. Does that make sense? Very powerful approach. So we're excited at the idea of developing dementia biomarkers. What causes Alzheimer's disease? Gosh, wouldn't we all love to know that, huh? What causes it? What is the top risk factor for Alzheimer's? Anyone? I'll give you a hint. It's right up here. Age. It's in the big red box. Age. You cannot get Alzheimer's without age. But you'll see up on this list, there's other things that you may or may not be aware of. Female, like we talked about earlier. ApoE4 genotype. This can be done. We can all do 23andMe and be told exactly whether you have various Alzheimer's risk genes. Hypercholesterolemia, I think this one's a little debatable. So this is a list not based on solid data, but based on what people think is currently in the field. And so I'd say of everything on this list, the most solid are diabetes, head injury. Head injury is the same as traumatic brain injury, and you've seen this in the news quite a bit, right? There is a strong link between brain injury and Alzheimer's disease. They call it CTE, but it's almost indistinguishable from Alzheimer's disease. So, okay? And smoking is up here as well, although I think that's also a bit controversial. All right, here's the map we were talking about. Can anybody tell me, we'll open up just this one slide, so we don't, just this one slide. Can anybody tell me in the audience why Alzheimer's disease is not evenly distributed through the United States? This map is based on blue being lower incidence of Alzheimer's, red being the highest, and then pink being somewhere in the middle. So this is all normalized to age, by the way. Does that make, because another example, for example, Florida has a higher elderly population. That's not taken into account here. This is all normalized to age. So why wouldn't, in the US, we develop the same amount of Alzheimer's disease per age group across the board? What does anyone think? What could we attribute this to?
Chemicals.
Chemicals, okay. Chemicals. And I think I heard environment. Did I hear environment? Okay. And so I agree. I think that there's an environmental component that we don't quite understand yet. And so there's a big push to understand what in the environment is creating inflammation. And there was a recent study showing that living near highways, because of diesel exhaust and things like that, there's a certain radius away from a highway that could put you at risk for Alzheimer's disease. So all of this is happening now. 5 years ago, the environmental link to Alzheimer's disease was essentially not there. But now there's supporting evidence of that, okay?
Seth,
Yeah.
you have about 4 colors, different colors in there. Which are heavy and which are...
Yeah, maybe it's not obvious here. There's some red states here: Washington, Arizona, Louisiana, Tennessee, South Carolina, Alabama are all the high incidence of Alzheimer's states. And then the low incidence, for some reason, Hawaii and New York. I can't think of what they have in common. Are the low incidence. Yeah, Florida.
I would think that they would be pretty high because a lot of old people go to Florida.
And you have to take this with a grain of salt because other factors have not been normalized here. So socioeconomic status and a lot of other variables could be involved here that are not taken into account. So, but it still illustrates an important point that something is happening unevenly across the country. And I was shocked to see this. Head trauma. So what happens during head trauma? Does anybody, aside from football, there's been a sport in the news a lot lately, soccer. Repeatedly taking 60 to 90-yard goal kicks on the head on the soccer field on a daily basis is probably not a good idea. And that's being shown, particularly Italian soccer players who were notoriously aggressive in their soccer playing days. And so I think there's an example, I don't have it on this slide. There are several very well-known Italian soccer players who have all gotten, many of them have come down with Parkinson's, Alzheimer's, or ALS. It can affect all of those diseases. So essentially what happens is these neurons, when they get stretched by the impact of the ball, these neurons get stretched. And when they do, they get essentially pulled apart. And that's what you see here with these little bulbous dots, similar to what I showed you earlier. I'm not sure how I would feel if my kids wanted to play football. I'd have to think about that. Actually, they're thinking of moving the age for football up from single digits, 8, 9, up to teenage years because of developmental issues that can happen from playing football. What'd you say?
Cheerleading, yeah.
Cheerleading. Oh, okay. That's interesting. I hadn't heard that one. Cheerleading. It's the best time in history to be doing Alzheimer's disease. Every time you look in the New York Times or the Philadelphia Inquirer, you see a new study. Here is a study that showed gum infection is related to Alzheimer's disease. So my very anxious mom called me and she said, I've had bad gums my whole life. Am I going to get Alzheimer's disease? And of course I said no, but then I looked into it a little bit more and there could actually be a connection between periodontal disease and Alzheimer's disease. It's a little complicated though because periodontal disease could impact the heart. There's some evidence that there's an infectious component that can cause heart issues, and you know the heart's connected to a healthy brain. So there's potentially circular argument there of which one's causing which. Poor sleep. Does everyone get at least 8 hours of sleep a night? I try to. Doesn't work out too well. I have good intentions going into my night's sleep. Pollution and herpes virus have all within the last year or two. Herpes virus is an interesting one. Of course, my anxious mom called me up when this one came out, and she said, you know, I have mouth sores and various things that are attributed to herpes virus. Am I going to get Alzheimer's disease? And so I think that it's very controversial right now whether infection can actually lead to the disease versus a person that has the disease being more immunocompromised. Those are two very big differences, very important. Every brain of a patient analyzed has some elevation in infection. That doesn't mean it caused it. So that's kind of a controversial thing that's ongoing right now. The peanut butter test. Has anyone heard of the peanut butter test? Oh, you're all going to go home and smell peanut butter.
Can't smell it, it's a sign of dementia.
One of the first things to go, in Parkinson's more so than in other diseases, is this ability to smell peanut butter. And I love peanut butter, and I can smell it from a mile away. And so if you have a very strong scent for peanut butter, apparently that's a good thing. And so they're actually using this as a biomarker. And so you can come into the office, there's a palette of smells, and you can go through the smells, and one of them is peanut butter. There's several others, and you can get a signature of what your smell is. Apparently the olfactory neurons degenerate in Alzheimer's disease and Parkinson's, and so you can use that as a biomarker. All right. What can we do about it now, right? We want to know what we can do about it now. It turns out our grandmom, who always used to say to us, go outside and play, to this day, physical exercise is really all that's been definitively, without a doubt, shown to be protective risk factor for Alzheimer's disease. So 30 minutes, 4 times a week, improved cognition, and decreased levels of tau, neurofibrillary tangles, compared to a stretching-only group. So that beautiful pool that I passed on the way in, that would be a great motivation to use that pool. Cardio is really one of the best things you can do. And we're not talking about running a marathon here. This is just mild cardio, 30 minutes a day, 4 times a week. To this day, I don't think there's as strong evidence of any of the other things on this list for being protective. A lot of people ask me about foods. What foods should I eat? And I would say that there's really no evidence right now that strongly supports a particular food. There is evidence if you have a healthy diet with mostly plant-based foods that your cardio and your heart health will be stronger. And whether you're directly impacting the brain or doing it through cardiovascular methods is not clear, but it certainly can't hurt to have a heart-healthy diet. And so what does that mean? Well, there's lots of things listed here: pomegranates, broccoli, a plant-based, high leafy green diet, a Mediterranean diet. These are all things that have been implicated, but I can't vouch for any of them scientifically because it's not quite there yet. Other things, triggers to avoid: allergies, obesity, oral periodontal disease, diet, sleep, smoking, stress, vitamin D deficiency, all implicated. Again, it would be hard-pressed to find a doctor or a neurologist that would say that any of these is definitely linked to Alzheimer's disease. One of the things that we found was that neuroinflammation is rampant in the brains of Alzheimer's disease people and mice. And so these are the things we think are all linked to inflammation that we should avoid. Now, these are not foods, but they're lifestyles. Aging, well, we can't do much about that, right? Or at least right now. Viruses, how do you avoid viruses? Well, do the logical, I assume, and stay away from anything that could be contagious, things like the flu, things like pneumonia. So we want to try to avoid being overly infected. Brain injury, pollution, smoke, toxins, and etc. And so these are things that if you adjust a lifestyle, you can potentially protect your brain. So we have a plan at UNC that's being built as we speak. We're physically building a center for neurodegeneration at UNC. And so this center is going to do the following things: address why our brains develop Alzheimer's disease. We're going to use that clarity technique to peer deep inside brains. We're going to slow down this process by identifying the genes and drugs that affect this. And we're going to do a lot of this in mice and rats. And we're going to ask these mice not to find their bedroom or find the keys that they lost, but we're going to ask those mice to find hidden platforms or to remember where they heard something or where they saw something, which is really, really good way of testing spatial memory. We want to better diagnose Alzheimer's disease in its early stages. We don't care what happens later. Not many people do. At that point, you've lost a third of your brain, right? We want to identify those things that happen early. So how do we do that? Well, we're going to bring together clinicians and researchers. We're going to identify biomarkers, and we're going to have people undergo MRI to see if their brain changes its size. And we're going to have them undergo PET imaging to see if we can see your amyloid plaques and your neurofibrillary tangles. And all of this is in place at UNC, and it's being built right now. And we're excited that in the next year or two, we're going to see a fully functional center for neurodegenerative work. Okay, and so this is essentially what I just said. We're bringing together 3 cores. This is very important because I think it's important for all of you to know what's happening in your backyard. This is just a few miles down the road, and we're going to be bringing together a very strong imaging core with a neurology program and a very strong research component to try to tackle this disease. There's no way to do this without these three things. If you're missing one of these, you're going to be behind in the game. And we want to make sure we cover every aspect of both curing it and identifying it. And that's how we're going to do this. So I'll end here. I'm sorry if this went way too long. I'm happy to take more questions, but I think I got to the end. And please support our mission. Okay, so this is my information. I also have a card and some material up here. But these are the points I want to leave you with. Be aware of what's happening right here in your backyard. Support our work and our research effort. And my feeling is that if you dedicate yourself to a disease, you can cure it. I think that can be said for HIV. Back in the 80s, it was assumed to be you were done. And now you can literally you can live a long, normal life. And I think there are a lot of people that were HIV positive that are legitimately HIV negative right now. And so we invested politically and we invested financially in HIV. And we've nearly eradicated it in this country. And so I think if we do the same thing behind this disease, it's interesting to think in 10, 20 years, we'll look back and think, well, why didn't we do that sooner? And I feel like 10, 20 years is a reasonable amount of time to find something that could be a cure. Spread awareness, be aware of all the great things we're doing, and be hopeful. Be hopeful, because there's a lot of great things on the horizon. This is the best time in history to be doing this kind of work. And so these are all the people that make it happen. I have a lot of great collaborators, especially this one. And she's getting her exercise, her 30 minutes of exercise. And I think that if we put our heads together, we can make this happen. And I'll stop there. I'm happy to take any more questions. Thank you so much for your time. That was the question on which supplements to take.
Supplements, and then there was something that you need a prescription for, but it said if your doctor doesn't give you this particular prescription, it's glucose, G-L-U-T-H, something like that. Yeah, I mean, it's my impression that there's very poor connection right now between a particular vitamin or a supplement and it being beneficial to Alzheimer's disease. And so now caffeine has been in the news quite a bit lately. If anybody's a coffee drinker in here, caffeine appears to boost memory. But to say that caffeine is going to help Alzheimer's disease is a bit of a stretch. Yes?
What would we look for in our mate or whatever that indicates the potential for those? What are we looking for? Someone that puts their keys in the refrigerator, or I forgot your name. When do we have an idea?
I think it depends on that person's history. So if you had a person that is someone that would never put their keys in the refrigerator and all of a sudden they're in there every night for a week, that would probably be concerning. And so I think you have to base it on that individual's history as opposed to, I mean, I've been forgetful lately, but I work all the time. And so I hope that that amount of stress from work doesn't cause me to start becoming more and more forgetful. But just keep an eye on the history of that particular person. That would be my suggestion. Yes?
Todd, in your presentation, there is about nothing that you mentioned that doesn't lend itself to be a danger for Alzheimer's disease. Like, you didn't leave out anything. Everything is potentially going to lead to Alzheimer's.
So are you...
Basically saying you guys got a ways to go yet.
We have a ways to avoid all of those things. If I leave you with anything, it's avoid all those things. Right here, yeah.
You mentioned several times brain trauma or brain injury.
Mm-hmm.
Would a brain bleed be considered a trauma to the brain?
I think it depends how the bleed was inflicted. If it was inflicted from a traumatic event, a single fall can cause a brain injury that leads to CTE, which is the clinical diagnosis. But if it's a bleed due to something else that wasn't a trauma, like a blunt force, then it sort of depends on how that was inflicted. So war veterans can be exposed to blast trauma. That's a trauma that can cause CTE. So there's different types of trauma, and depending on the amount of force exerted will determine the impact on the brain. Yeah.
How much research into the other dementias are you doing at UNC?
We work on FTD, frontotemporal dementia, and so between FTD and Alzheimer's disease, that covers about 75% of all the dementia. But there are other types of dementia, like vascular dementia and Lewy body dementia, that are sort of separate clinically. We don't work on those as much. We're mostly working on things that involve these tau tangles because we think we can get rid of them. And so FTD and Alzheimer's disease have these tangles. So that's our passion, to get rid of those.
That's interesting.
Yeah.
You have an association with the Alzheimer's, you have an association with the Alzheimer's clinic up in Wake Forest?
I don't, but I've had lots of productive discussions with them. We'd like to collaborate with them. We don't have anything yet. Maybe some of it is because of proximity, and they're a little further out than places like, we do work with Duke and NC State, so maybe Wake being a little bit further away hasn't really brought them into the mix quite yet. Yeah?
The mass spectrometer work you talked about,
Uh-huh.
you identified all those proteins very precisely, you said.
Uh-huh.
What's the relationship between all of those proteins that you identified and the protein content of those tangles?
Well, the tangle is thought to be made almost exclusively of this one protein, this tau protein. And so the protein that makes the tangle is almost one protein. It's this protein called tau, T-A-U. You may have seen this in the news. Now, the proteins we're identifying on the mass spectrometer could be any protein at all that happens to be in the blood. And that it changes with disease progression is the key. And whether it's in a tangle or not is not really what we're going after with that experiment. Yeah.
So, obviously, there's no way to dissolve that tau protein with proteolytic or F-to-Dase activity?
Well, in a brain, very difficult, in a live brain, to dissolve it. And you might think, why can't you just chew it up? Why can't you just... The problem is finding it, and then the treatment to get rid of it would injure your normal brain tissue. So we're really good at degrading it, and when you get it out of the brain, we're really good at that. But that's not as helpful. We want to degrade it in the brain, which is why we're using gene therapy approaches now to deliver things to the brain that might get rid of these tangles. And so this is now, it's not something I've talked about, but maybe when I come back at some point you can hear about all the gene therapy approaches that we're using. We're very excited about that. Any other questions?
Well, I asked you before about amyloidosis and correlation, because I know there was some research being done by someone I knew at University of Tennessee about amyloidosis, and he was talking about a correlation between that and Alzheimer's. So
Right.
I just wondering if there's any relationship between those.
Yeah, I think now it looks clear that the amyloid part happens early in the brain, and that when you're in your 30s, say you get Alzheimer's disease when you're 75, it seems that 3 to 4 decades prior to that is when the amyloid part is happening. So that's a long time. How can anybody even conceive of what's going to happen in 40 years? So say you get Alzheimer's when you're 75, if you could just rewind time, you'd want a drug that would dissolve the amyloid part 35 years prior, so when you're 40. So I just turned 40 not too long ago.
You're a baby.
If I was going to get Alzheimer's when I'm 75, I should start taking a drug right now. But anybody would think that would be hard to believe. What drug would I take? How do I know that I would get it? And would I want to take a drug for 35 years? I don't know that I would even want to. Maybe I should take metformin. Maybe not, yeah.
What you're saying is it's very optimistic, but there are, how many researchers like you are there out there? How many different labs are there out there doing comparable research?
There are a lot, but we're the best one. So that's my direct answer to that question. That's why I'm here, I guess so. You picked the best. Thank you, guys.
Could I speak about hydrocephalus?
Yeah, sure.
I had an experience with this. My sister, a little less than 2 years ago, was slurring speech, mixing up words, falling about twice a month, and having bladder issues. Those are the 3 key symptoms, and they found hydrocephalus in her brain. And she had to fight. We had to go from doctor to doctor to fight to get the MRI to see the fluid on the brain like a baby. And she had a shunt put in, and she's fine. So there's one brain condition we can fix.
That's a very good point. That is a good point. As long as you keep shunting, this is a condition that is fixable. Have a good night, everyone. Thank you so much.
Thank you.